Hello, my name is Mary Garcia, and my topic for my modeling project is phototransductions of the rod cells in the retina. So I'm going to start with phototransduction, which is basically the process by which light energy is converted into a greater receptor potential. And one of my second terms would be basic units of visible light, which, can you guess, is obviously photons. Photons are very important in the phototransduction functions and cycle. Moving on, we have osmin. So basically, osmin is a protein that is found in the rod cells, and it's a photopigment that is the photopigment protein. So when you have osmin and retinal pigments, you should normally see that retinal changes shape, also meaning that vitamin A is presented in this cycle. Okay, so retinal layers. So to start off, retina is the third layer inner coat of a nervous tunic which lies the posterior 3/4 of the eyeball. So our three layers would be number one, photoreceptors layer, and rod and cone cells specialized to transduce light in this first layer, which is the photoreceptor layer. Then we have bipolar. Cells in which bipolar layer, which we have established pathways for horizontal cells, and horizontal cells are they transmit inhibitory signals to bipolar cells, so they inactivate with each other, and both bipolar and apocrine cells produce graded potentials to ganglion cells, which depolarize and initiate action potentials. And ganglion layer is our last layer, which is basically neurons whose axons merge to form optic nerve. So basically, you already know that optic nerve is found in one of our 12 cranial nerves, which is basically in one of our foramens, which is in the optic canal. Sorry about that. So that's very important because that is what forms ganglion cells, is what forms the optic nerve. So we're moving on into rod cells and cones. Exactly. What I have for this is basically rod cells are major photoreceptors, in which one fun fact that I have is that we have about 125 million rod cells in our eyes just to be able to see color and just be visual. They are also highly sensitive to light, and they can therefore see in a dark room. Although we're able to see in a dark room, it does not mean that we're going to be able to see the same objects and same colors as we see during the day, where we're exposed to light. So, moving on, number 5, which is basically this is what we call hyperpolarization and depolarization. And hyperpolarization is in the absence of light of the sodium channels, which stay open due to binding of second messenger cGMP, which is cyclic guanosine monophosphates, which causes to depolarize, keeping the potassium channels open at the synaptic terminals, which produce neurotransmitters that inhibit bipolar cells. Basically is the same thing. This is what hypolarization and depolarization do. It's basically, hypolarization is basically on the light absorption. So we are moving on. We have G protein. G protein is also known as transducin and is found in the disc membranes of the photoreceptors, and also initiates the activation of cGMP. So we have another similar term in which is kind of similar with the name, however different functions and different meanings. cGMP phosphodiehydrase is basically an enzyme found in the cells that convert cGMP into GMP, which is basically guanosine monophosphates. And I have one of my cones visual picture that I made to show you guys, to be more understanding what cones look like and what they have and the 3 types of colors they have in them. Basically, this is what it is. We have our structure, which is, this is the cone, as you can see, the cone. This are the discs. This is, we obviously have a nucleus, and this is our bipolar cell. And the 3 types of colors it has red, it has green, and it has blue. So this is what we have in our cones. As you can see, it's very different from our rod cells, which this is what our rod cells look like. So it's very different. The cones and the rods are very different. Okay. So, rhodospin, it is a hymodimeric protein that contains high G protein coupled receptors found in the membrane disc of the outer segment of the rod and the cone cells. Also known as visual purple, which is sensitive to red light and found in external segments of the rods. Basically, I have a 3D model also to show you a little bit depth in how it looks and what it looks like. So this is what I have. This is my model. As you can see, this is rhodospin. This is ospin right here. As you can see, this little red across. That is what we call the retinal. And these are the ospins. And this is the whole structure together, which is basically the rhodospin. Okay. So, moving on. 11-cis is very important because it activates ospin, so it's very important in the structure in order for us to receive everything and function right in the phototransduction cycle. We have to have 11-cis. As you can see in the first picture, it's very active in there. It also it absorbs light and changes liner to make 11 transform. Alright, so GTP, which stands for guanosine triphosphate, very similar to ATP, and it's a high energy compound like cGMP. And we're moving on because that's basically the most accurate way to define these and make them more simpler. Voltage gated potassium ions are transmembrane channels just for potassium, and any channel opens and closes to change electrical potential across the cell membrane. So this is what it is. Moving on to nothing but not least, this is what I have for calcium ions and potassium ions, which basically calcium ions trigger exocytosis of the ACH into sympathetic left, and potassium ions are positive ions found within the cells. That's what basically it is. My last term of the phototransduction in the rod cell Britannia is protein kinesis, which is activated by cGMP, which you guys remember is cyclic guanosine monophosphates. And I have one more last picture that I want to show, and it's basically a 3D model of ospin, just to show you a little bit deeper how it looks. It's not the exact color, but I chose these colors to be vibrant. Which is this? These are like the ospin 3D. This is how they look inside the rhodospin. As I showed you, this is what is in here, and this is my little 3D model and how they look like inside. And that is it for my modeling project. Thank you very much for watching, and I hope that this was very useful and efficient for everything. Thank you.